Hello guys and welcome back to Excelsior Academy. Here I am Dr. Ruby Joshi with again a very important and interesting question. So as I used to say before, we know the concept is just that how we are going to apply to give the answers at the interview. Okay, so let's see the question. Incorporation assay measures the DNA synthesis using 32p labeled DNTPs. So guys, in order to answer this, you need to know what is incorporation assay. So I have already taken this as the lecture number 2 in the Watson summary series, chapter 9, DNA replication. Please go through that lecture before answering this. If you have watched that lecture, then it is easy for you guys to answer this. ठीक है, और अगर आपने नहीं भी देखा है, still using your logic and basic concept, you can answer this. Okay, so if you use, so our first question is, if you use DNTPs labeled at this is beta and gamma phosphates, you do not detect any radioactivity in the newly synthesized DNA. Now this is happening why? ठीक है, ये बहुत simple है. See guys, whenever DNA is getting synthesized, what happens is that whenever new nucleotide is coming, you know the chemistry behind the DNA synthesis. Remember the SN2 mechanism. You have to remember all this thing, okay? This is the basic chemistry behind your DNA synthesis. What is happening in this? So there is a 3-OH which is attacking at the beta phosphate, okay? So what is finally having? The 2 phosphates are removed as a pyrophosphate. So what is remaining? Your DNTP is becoming now for example, you have got the one pyrimidine, one base is there, वन फॉस्फेट इज देयर एंड द राइबो शुगर. ठीक है, तो कौन सा फॉस्फेट हो रहा है आपके पास? अल्फा फॉस्फेट इज रिमेनिंग इन दी डीएनए सिंथेसाइज स्ट्रैंड. ठीक है, और आपको जो बीटा गामा फॉस्फेट है, दोज आर रिलीज एज योर पायरोफॉस्फेट. Okay, so they become the pyrophosphate. Now this is later hydrolyzed and it is never incorporated into the growing DNA chain. Okay, because again it is hydrolyzed and this becomes 2 inorganic phosphate, which is then releasing very good amount of energy in order to catalyze or help in the carrying out the further reaction. ठीक है, इसलिए हमें कभी भी उसमें radioactivity नहीं दिखाई देगी because beta and gamma are never present in the newly synthesized DNA. So guys, I hope this is clear to you. ठीक है, बहुत ही सिंपल था, इसके लिए आपको किसी इनकॉर्पोरेशन ऐसे की जानने की भी जरूरत नहीं थी. Now let's go to the next question. Following incorporation of the 32p labeled DNTPs, you must separate the unincorporated labeled DNTPs from the newly synthesized DNA strand before measuring the amount of the 32p incorporation. Of course, ठीक है, तो इसमें क्या बोला है कि labeled and unlabeled को हमें separate करना है. Now, explain how gel electrophoresis serves as a means to separate the unincorporated 32p labeled DNTPs from the newly synthesized DNA strand. So guys, this is really very simple. Electrophoresis का काम क्या होता है? Gel electrophoresis का क्या काम है? हाँ, so we know that in the gel electrophoresis it is going to separate based on the size. अब हमें मालूम है जो synthesized DNA होगा उसका size ज्यादा होगा as compared to the unincorporated 32p labeled DNTPs. वो तो कुछ ऐसे रहेंगे. Now अब क्या होगा आपके जेल में? The moment you are going to load these 2 samples, what will happen? The DNA which has been synthesized is going to show you as a band, whereas the unincorporated 32p labeled, they are just the small molecules, so they are going to run faster in the gel electrophoresis. Okay, maybe they might pass the gel also. So क्या होगा at the end when you're going to visualize the gel, what will happen? Only the newly synthesized DNA it is going to show the band, whereas the unincorporated 32p labeled DNTPs, they will be washed off. So this is how you're going to use gel electrophoresis as a good mean to separate the unincorporated versus the newly synthesized DNA strand. ठीक है, and yeah, that's all. था ना simple? तो इसी तरह से आपको सारे आंसर्स देने हैं. ठीक है, मैंने हमेशा से बोला है जब भी आप कुछ भी एक्सप्लेन करना चाहोगे, इफ यू हैव दैट कॉन्फिडेंस, टेक द पेन एंड मार्कर एंड एक्सप्लेन देम विद द डायग्राम्स. ठीक है, जितना ज्यादा आप अच्छे से समझाएंगे उतना ज्यादा कॉन्सेप्ट आपको भी क्लियर होगा एंड सामने वाले को पता चलेगा कि दैट यू रियली नो योर कॉन्सेप्ट्स वेरी वेल. So guys, that's all for today. I hope you're enjoying the series and soon I'm going to start with the other topics as well also. So stay tuned with the Excelsior Academy. Keep watching and stay at home, stay safe, study with us for your next target. Thank you.